everyone. I'm Andres. I am not part of the Bioclock Consortium because I came to the Netherlands after the consortium was founded. But anyway, I'm very happy to be here. I have attended the last 2 meetings, but never got a chance to speak, so I'm grateful for the opportunity. So to start, so I can introduce myself to the consortium, I wanted to tell you a little bit about my not so direct path to assistant professorship, but still a path. So I started as a circadian biologist during my PhD in the city of Quilmes, that's in Buenos Aires, in Argentina. I did my PhD with Zhao, and that was with animals. So I was working with animals at the time. But then, in my postdoc, I transitioned to Romanowski's lab, who is a plant circadian biologist. So I moved from animals to plants. From there, I jumped through the Atlantic to Scotland, where I got extra training in photobiology in Karen Halliday's lab. With Marie Curie, I made my way to Utrecht, and after that, I started as an assistant professor, and I'm now in Wageningen University in the molecular biology chair group with Ronald Pearce. So, got training in photobiology, lower left of the slide. Then during my postdoc, I started doing some training with bioinformatics. Circadian biology all the way to the top. Then bioinformatics and then photobiology. But in addition to my research, I also had other hats. I was also teaching, since even before I started my PhD, so I started teaching in 2007. And then I was also involved in changing the research culture of the different institutes where I was at. For example, as an open research advocate, now I serve as a Wageningen data steward. And I also participated in several equality and diversity committees, and I'm currently acting as an external expert member for the Edinburgh Plant Science Network. So what are circadian rhythms? I guess I don't need to tell you this, but we live in a world that spins around these axis with a period close to 24 hours, that leads to the fluctuations of days and nights, and several other daily variations in environmental variables, and this has shaped the evolution of most of the organisms that live in this. This led them to evolve an internal timekeeping mechanism that can predict those changes before they happen. And this is called the circadian clock. So where did it all start for me? So my PhD was in circadian rhythms in C. elegans. When I started, this was the first C. elegans lab in Argentina. So we were starting with a new model, and also a model that we really had no information about its circadian system or whether it actually had circadian rhythms. So my PhD was about stress tolerance, abiotic and biotic stress tolerance. So I described the first rhythms in osmotic and creative stress. I also helped with the testing of a activity tracker, but this device that you can see here. So it has some lasers that allows you to track the activity of C. elegans as it flashes in the liquid culture by interrupting an LED system. And then we also describe rhythms in feeding, defecation, oxygen consumption, melatonin synthesis. We then found a role for PDF, so for the homologue of the pigment effect dispersing factor from Drosophila. We also found it in C. elegans. We did some bioinformatics searches to try for clock components homology. And in 2016, when I was already a postdoc in Marcelo's lab, I helped develop the first luciferase-based report for C. elegans. But then I moved to plants, and then people started asking me, wait, why did you do that? You were working with animals, why transition to plants? Well, I don't know if many of you know, but we usually remember the year 1729 as the year when chronobiology was born. And this was because there was this French astronomer, Jean-Jacques Toussaint, who was working with a plant called Mimosa pudica that opens its leaves during the day and then it closes them during the night. But that was not the most important observation that he made, but it was actually that if you grab this plant and you put it inside of a cabinet, in the absence of external cues, these rhythms of opening and closing of the leaves, they continue. So this makes me think that there was an endogenous timekeeping mechanism that allowed the plant to sustain these rhythms. After 1729, then in 1751, Linnaeus made his floral clock. So it's a clock that has different flowers that open at different moments of the day. And then it was not till 1920 or 1930 that Boone proposed that rhythms could be inherited. So there was a genetic component to circadian rhythms. Yeah? He also proposed that this internal timing mechanism could also help an organism understand seasonal rhythms. We also know most of the clock components of plants, only that the difference in the timekeeping mechanism in plants is that it lacks many positive elements. It's basically a succession of repressors that repress one another. That's called a repressivator. Although, since this model was done in 2012, some positive elements were also found. And the latest model, so this keeps changing, so even though we know a lot about the clock, this model keeps being updated, and the latest one was published about a month ago. Yeah? And this one uses absolute RNA quantification, yeah, to get you absolute values of protein levels afterwards. Feel free to check it out if you're interested. We also know which cues can entrain the circadian clock, such as, for example, blue light, temperature fluctuations, photoreceptors, and even sugars produced by photosynthesis. What else do we know from plants? Well, that the clock controls several processes, as it does in other organisms, such as, for example, chloroplast movement. Yeah? So the chloroplasts move from one plane of the cells during the day to capture more light, and then during the night they move to the sides of the cells. We also know that they're involved in stomatal opening, yeah? Which is the stomata being opened by a morning element of the clock. Yeah? Hypocotyl length, yeah? Cotyledon and leaf movement, petal opening, and also seasonal outputs, like for example flowering. And of course, gene expression or protein phosphorylation, cytosolic acid levels. We know that the clock controls this, but for many of those, we also know how that happens. Yeah? So, for example, in this, on the right panel, yeah, what you can see is the clock elements here in the center, yeah? And then the targets, and then the downstream processes that are controlled by the clock. And many people here might wonder, how do we characterize circadian rhythmicity in plants? Well, the classical way to do it, and the easiest, is to look at leaf movement. Yeah? So you entrain your plants, yeah, to whichever photoperiod condition you want, and then you transfer them to constant conditions. And here we also have another difference with working with mammals or with insects. Yeah? Instead of using constant darkness, we use constant light. Why constant light? Yeah? It doesn't lead to rhythmicity in plants, and the plants need it to continue producing sugar, yeah? So to produce their food. If you keep them in darkness, then you need to add exogenous sucrose to keep the plants fed. What we see here is in the y-axis the leaf angle, and in x the time since transfer to continuous light. And you can see how beautifully they flap around. The other technology that we use a lot is whole plant tissue bioluminescence. So we can also grow these plants in 96 well plates, when they're super tiny, so as seedlings. We add luciferin, and then we can track. We can track them in entrainment conditions, and then transfer them to continuous conditions, or do any sort of treatment. We can see that. And through devices like the nightshade, we can even get images of these plants as they're growing. Before any one of you asked me in the coffee break or at lunch, I do not have this device. I don't have a nightshade, but I wish I did. Okay, so this was just a brief intro to plants, so you could see the differences with whichever organism you're working with. So how was my transitioning into plants? I told you I went to Marcelo's lab, and there I worked on many different projects, but my main one was how the clock controlled alternative splicing. So it was a good project to transition into, because it meant that I didn't need to know so much about the husbandry of the organism, because it was mainly bioinformatics, and it was also going to train to get bioinformatics training at the time. But I also worked on how light affects alternative splicing, the link family of genes, so these links are part of the positive elements of the clock, and also how bacterial infections could disrupt the clock for their own benefit. So when I got to Marcelo's lab, we knew that the clock controlled many co- and post-transcriptional processes, such as transcription, nuclear export, polyadenylation, mRNA degradation, and we had some indications that alternative splicing was also controlled by the clock, or that alternative splicing could affect some of the clock components. But what we didn't know was to what extent did the clock control alternative splicing. And just as a refresher, but what do I mean by alternative splicing? Well, it's this process, where from a single gene we can have different gene products through the combination of different modular components that are called exons and introns. And some of the different types of alternative splicing events are the constitutive one, where the intron components are removed and only the exons are retained. Exon skipping, the second, where one of the exons can be completely removed. Intron retention, the name says the intron is retained. Or alternative splice sites, either on the 5' side or the 3' side. We also have more complex events, but I'm not going to go over those. So the question was, what is the extent of circadian regulation of alternative splicing? So that led us to, from Argentina, to the first Arabidopsis global circadian mRNA-seq. Yeah? I was surprised that no one had done it by the time that I published this. But we did. So at the time we had a lot of microarray information, but no RNA-seq. So how did we do this? So we grew plants in plates, as you see in panel A. Yeah? We subjected them to 12 hours photo periods of 100 microinstates, which means 100 micromoles per square meter per second of light. And when I give that measurement, that means of light between 400 and 700 nanometers. And then after 12 days, they were transferred to continuous light. Yeah, remember, these are our continuous conditions. And after 24 hours, we started sampling every 4 hours. Then we did prepare the libraries, we did the sequencing, and voila, lo and behold, on the left-hand side, we can see a heat map, graph gene expression in Arabidopsis, and we found a total of 9,127 genes per cycle. And we used JTK cycle to obtain these genes. And we can see a nice spectrum of different phases. Yeah, in panel C, there's a histogram of acrophases. Yeah, and we can see that there's two distinctive peaks, one's around dawn and another one around dusk. Yeah, but there's a nice spread of other phases as well. And we quantified by qPCR just to validate our approach known clock genes, such as CCA1 or LHY, which are morning elements, DRR9, also morning element, and Reveille 8, which is one of the positive elements of the clock. So using this approach, we found novel circadian genes that were not previously described to cycle, mainly because many of them fell outside the microarray technology. They were just not there as probes. And we could see that these new genes had to do a lot with RNA processing, RNA metabolic processes, non-coding RNA, which was great because we wanted to work on splicing. So that's nice. And we also found 146 transcription factors that were not known to cycle. And that's nice because that also opens new avenues to work on circadian research afterwards. They're published, they're there for the taking, but this was not our main answer, our main question, sorry. Our main question was about the extent of clock control of alternative splicing. So, can we do that? So, the task proved a little bit more challenging than we originally thought, because when you use these RNA-seq technologies, you have read lengths of around 100 base pairs, and that means you have to reconstruct the isoform. So instead of doing that, and running the risk of reconstructing a wrong isoform, what we did is instead of using an isoform-centric approach, we use a splicing event approach. What does that mean? That we bin the genome in small discrete places using the genome coordinates, and we assign those bins as being an exonic bin or an intronic bin. And that allowed us to free us from this isoform reconstruction step. And with that, yes, using JTK cycle, we could analyze this. And this also led to the development of an R package called ASPLI, which is alternative splicing pipeline, that you can also use with your own data sets to analyze splicing events. And this was done by Stefania Mancini, who was a PhD in Marcelo's lab at the time. So to answer the question, does the clock control alternative splicing? Yes, it does. It's not as widespread as I would have wanted, but the facts are the facts. So what we found here is that there are 1,513 clock control events, which we name CCEs, and this corresponded to 1,116 genes. And most of them were intron retention events. In mammalians, usually the event that you found the most is exon skipping, but in plants it's intron retention. And here you can see one such event on the right side. And if we look at the reidentity plot, we can see one of these events, where it's not there at the beginning, but as time goes by, then you suddenly see that increase in the read density in the intron, then it goes away, and then it comes back. So we said, okay, if the clock controls alternative splicing, then there must be splicing factors that should control the splicing events. And what we did is we intersected our own curated list of splicing-related genes with a list of genes that we termed the robust cycling genes. which are the ones that cycled in all the different data sets that were available at the time. And we found 31 of them that cycled. And one of those is this gene that's called splicing factor 30. Yeah? It was previously known as a survival motor neuron, so you know it. But using phylogenetic assays, we discovered that it's more closely related to splicing factor 30 than to SMN, so that led to the renaming of the gene. We tried to characterize some mutants. That's what you normally do in Arabidopsis. You work with 1 or 2 or even 3 different alleles of the gene that you want to study. Those are the ones that you see in the lower center. Yeah. And we did the leaf angle, so that technique that I showed you was the classical one of rhythmicity. We found no differences there. But then we also looked at photoperiodic flowering, yeah, which is a seasonal process that's controlled by the clock, and we saw that in different conditions, like continuous night, long days or short days, yeah, these plants were always early flowering. So then we said, okay, let's take a look at the events that are controlled by splicing factor 30. We wrote these plants in continuous night, yeah, to get rid of any rhythms from the start. We sequenced them, and then we found that there were 286 events that were dependent on splicing factor 30, and when we intersected those with the clock control events, we found 32 that were under control of this splicing factor. And then when we did qPCRs, yeah, to try to validate some of the events that were both clock controlled and also controlled by splicing factor 30, what we found is that this splicing factor does not disrupt the rhythm, but rather changes the absolute levels of the intron retention events. So it just moves the baseline. Okay. So, to summarize these feats, yeah, what we can see is that there are environmental cues, that entering the circadian clock are Arabidopsis. This leads to clock control of gene expression, and also of alternative splicing events. Some of them are shared, yeah, so there are some genes that are clock controlled and also have events that are controlled by the clock. The gene expression data is now available in a Shiny app called Caster, yeah, that was developed by a different group. And from these clock controlled events, we found a subset that are controlled by a splicing factor called splicing factor 30 that mainly affects these intron retention events by moving the baseline. So that was done in Marcelo's lab. Then years went by, onto new adventures, and now I can introduce my own group, which is called the Times Lab, yeah, which is timing of environmental signaling. And what are we interested here? Our main question is, how does a dynamic environment affect plant development? And we have the following questions. So, what, which means which cues and which stress? When, so is timing important for this cue and stress? Where, so is this tissue or cell type specific? And how and why? So, is it clock modulated or directly driven? And what are the mechanisms behind it? So mainly, we focus on all the arrows, but not the clock itself. So another message, yeah, people are key, yeah, for our labs. So I want to acknowledge all the past members from my lab, master students, and also PhDs, people that came from Spain just to do a short stay in the lab. Sharan, who was my first postdoc, yeah. And also our current people. So Chankun is my first PhD candidate, and she's currently working on how the clock can modulate shade avoidance syndrome in different photoperiods, yeah, so the seasonality aspect of shade avoidance. Then we had Florencia Bojino, who came from Córdoba in Argentina to work on the crosstalk between immunity-like signaling and circadian rhythms. And a recent PhD candidate, Nidhi Mishra, who is working with us on environmental modulation of protein mobility in plants. Oh, sorry. Yeah, that's it. Thank you very much. Thanks for your attention, and I'm here for your questions.